So now I'm inviting the first speaker, keynote speaker, Professor Ipsita Roy. Ipsita has earned her PhD in biochemistry from the University of Cambridge, UK. She is currently Professor of Biomaterials at the University of Sheffield, and she was previously Professor at the School of Life Science, University of Westminster, London. Her research interests include natural polymers of bacterial origin and their use in medical and environmentally friendly applications. She's skilled in biomaterials, tissue engineering, regenerative medicine, nanotechnology, green polymers, fermentation technologies, all very interesting topics. The title of her presentation is Natural and Sustainable Polymers of Bacterial Origin and Their Biomedical Applications. Please, Ipsita.
Thank you very much, Elisabetta, for inviting me and for your kind introduction. And good morning, everybody. I absolutely love Italy, and Milan is, of course, so beautiful. So really glad to be here and be part of a polymers conference after a lot of time. Normally, I'm attending biomaterials conferences, so I'm looking forward to hearing all the speakers. Just to clarify, in order to be on time, I know it's about 5 to the hour. Should I then finish at 40 past? Is that what you like, Elisabetta?
You have 40 minutes speech and 5 for discussion.
Okay, so I finish at 35 past then, and then 5 minutes. Okay, thank you again. And so as Elisabetta mentioned, I'll be talking on natural and sustainable polymers of bacterial origin and their biomedical applications. So these are the 4 main polymers that I use in my laboratory, with a large focus on polyhydroxyalkanoates and bacterial cellulose. Gamma polyglutamic acid and alginate are the younger members of the lab, and today I will not be presenting any research on that. So this is, you're all chemists, I'm guessing, or polymer chemists, so it's obvious that these are polyesters, polyhydroxyalkanoates, and as you can see, the R1 and the R2 and the X values are variable, which means what we are dealing with is really a family of polymers, not just one polymer type, and we mainly divide this family into 2 main types, the short chain length PHAs, which are normally hard and brittle, and the medium chain length PHAs that are very, very elastomeric in nature. So this is how I produce my polymers. We have a pilot plant in the University of Sheffield. This is about 30 liters working volume, and therefore we can really scale up the polymer production. Obviously, without having enough polymer, we can never do anything with them. So we aim to produce these polymers in large scale. So when I started many, many years ago, frighteningly long, about 25 years now, I wondered what I should do that is important. And I'm sure the younger scientists here, when you start your group, you'd wonder, what is the topic area that I should choose? I chose polyhydroxyalkanoates. However, there were 2 large groups, one in MIT and one at Münster, already working on these polymers. So what I thought was I would like to produce these polymers using gram-positive bacteria. And the main reason for that was that these polymers lack lipopolysaccharides, which are hugely immunogenic in nature. And I wanted to use these polymers for medical applications. You don't want an immunogen in your polymer if you want them for medical applications. So I thought, okay, let me start gram-positive bacterial production. The community was quite scared. They said, you can't break these bacteria easily. This is not going to work. It worked, and we got large quantities of polymer from this, and now there is industrial production using gram-positive bacteria. The other reason I use bacteria for polymer production rather than synthesis or using natural polymers from nature is the fact that I can completely control the process. It's not that I'm isolating alginate from seaweed, which has grown somewhere, I don't know what temperature, etc. So the monomer content can change. Molecular weight can change. And all of you are polymer chemists, and you realize a different molecular weight, a different monomer content makes a completely different monomer. a polymer. So this is the type of conditions that I control: dissolved oxygen tension, pH, temperature, every nitrogen concentration, every single thing is absolutely watertight, which ensures that every single time I produce the polymer, it has exactly the same molecular weight and monomer content. Extremely important for making things with these polymers. So this is, of course, making sure that I isolate this polymer from bacteria, making sure I get exactly the same polymer every single time. I do an NMR, proton and C13. I also look at GPC, gel permeation chromatography, to ensure that the molecular weight is also the same, because that's very crucial. So this, you can see, is quite a high molecular weight polymer, and the PDI is about 1.99, which is quite good, I think. These are the properties of the polymer, so I won't read out everything for you, but I'd like to, firstly, processing is important, so depending on the type of processing, the polymer property obviously changes. And if you look at the elongation at break of this polymer, it's quite low, so it's brittle, like I mentioned, and the Young's modulus for a polymer is quite high, in the gigapascal range. So this is the polymer that are mainly used for bone tissue engineering. I also produce the highly elastomeric. We have about 7 different types in our laboratory, and these are extremely elastomeric in nature. Again, a very controlled fermentation process is used for the production, and we ensure that the chemical structure is exactly what we want. So we do an FTIR spectroscopy, we do NMR, and ensure that we have exactly what we think it is. So the GPC, you can see here the PDI is even lower, about 1.76, and the molecular weight is slightly lower than the previous polymer, but quite high otherwise. Again, looking at the properties, you can see here the elongation at break is much higher than the previous polymer, about 290% and 558, depending on how you've processed them, but the Young's modulus value is much lower. So we have 2 types of polymers, one rigid and strong, the other very elastomeric and not that strong. I also use energy and sunshine, as you have in Italy. We don't have much of that in the UK, but I'm trying to use whatever we have for the polymer production. So algal production of PHAs is another thing that I've started with Jag Spandal at Sheffield. So what do I do with these polymers? Of course, make structures with them. And I must say that we are perhaps one of the few groups that can process PHAs very easily. And I guess because we've been focusing on PHAs and have been trying to make structures with them, we have learned along the way. So this is just to show you how many different types of structures can be formed with these polymers. We can use almost every single traditional methodology used for polymer processing for these polymers, which many people find it quite difficult to process PHAs. Not because it's difficult, because people aren't used to these polymers yet. So this is another overview. So we've got porous structures, microspheres for drug delivery. We gyrospin and create fibrous structures, 3D print them, make tubular structures, melt electrospin them. This is melt extruded and then a laser cut. And then we have 3D molded structures as well. Here's FDM printing, a fused deposition modeling printing of the medium chain length, the highly elastomeric, and you can see how elastomeric that polymer structure is. And this is again another, so like I said, I have many different types of medium chain length PHAs. This is yet another one where we created a slightly bigger structure because this was for bone. And this is PHB, which is the rigid form. Many people cannot print PHB. We have some tricks up our sleeves, and therefore we can print them. This is on its own, and on the right you can see it's a composite with a particular additive. So I've been talking about this. I don't think anyone in this audience, except for Elisabetta, has heard me before. I've been saying this for years. People initially to say, my God, what is she saying? This biological polymer, this is never going to work. But it's becoming reality. Sustainability is becoming very important. And in the medical world, there aren't many polymers that are sustainable yet. And it's very important we get to them very soon. So PHAs are coming really important. I am getting a lot of requests for these polymers from companies who need to now meet the green agenda levied by the government, and rightly so. So I'm not the first in thinking about medical applications of PHAs. Like I said, two groups. The MIT group actually spun out a company many years ago called Tepha. And they actually have FDA approval for one type of PHA, that's P4HB, that I'm not talking about today. But this is great news for us from the PHA community, that we can actually get FDA clearance because they're already familiar with this polymer. So what have I done in the last 20 odd years? I've used these polymers for a variety of different applications. And these are so biocompatible that I haven't found yet a cell type that will not attach and grow on these polymers. And I think that's a lot to say about a polymer that's produced by a bacteria. So today I will talk to you about a few examples. So bone tissue engineering is the first type of tissue engineering that I started with these polymers with actually a collaborator at Imperial College, Aldo Boccaccini. Some of you might know him. So today I will show you some examples. There's lots of bone tissue engineering work we have done. This is a P3HB, that is the rigid form of the polymer, along with hydroxyapatite, which some of you might know is a bone regenerative material. So we mixed them up and gyrated or gyro-spun them as a pressurized gyration. And this is the process. If you, I don't know if you've had candy floss while you were a child. This looks very much like candy floss, actually. And it is remarkably porous and fibrous in nature. Wonderful for cells to attach, grow, and nutrients to be supplied in the tissue that you develop. So I also love animals. So before going into in vitro models, we use what is called a CAM model, which you take an egg, and where a chick embryo is developing, and put your polymer on top of that. See whether it kills the chick or whether it results in any vascularization, which is very crucial in tissue engineering. Always a challenge. And indeed, we got red means vascularization, blue means collagen formation. We got both. And that told me that, yes, this material is indeed worth taking forward. So then we went into animals, and then we used a rat. And all these panels show different types of groups that we used of the material. The last one proved to be the best, where we had P3HB, hydroxyapatite, and STRO1 positive cells. These are bone cells that we added onto our construct, and that led to better vascularization and regeneration. I move on to another example of bioactive glass. I'm sure you've heard about these glasses. This is a zinc-doped borosilicate-based bioactive glass produced in Erlangen in Professor Boccaccini's lab. And we created composites of this with B3HB with different volume percentages. And we saw that the composites that were formed were very uniform, a good, nice distribution of the filler. We also looked at the presence of the various elements, and this is SEM-EDS, and we saw all the expected elements that we thought should be present in the composite. And of course, then we look at the mechanical property. A composite's mechanical property is perhaps the most important property, and we got nice high Young's modulus value with the addition of the borosilicate glass, 20%. It gave us a higher Young's modulus, a similar elongation at break, and an ultimate tensile strength increased as well. This is exactly why we wanted the composite. We also wanted bioactivity. So this is simulated body fluid, just like the fluids in our body. We can simulate that fluid and get and soak your sample into simulated body fluid and see where the hydroxyapatite forms, because bone is hydroxyapatite and collagen. And what we observed, all this irregular material that you see on the surface is indeed calcium phosphate forming, and this told us again that this material is indeed promising. Another very important property that I looked at was antibacterial property. We all know that many patients suffer from sepsis when they undergo these operations, and especially elderly, immune compromised. There's a lot of mortality because of sepsis. So if the construct itself can be antimicrobial, that will solve a lot of that problem. So we tried to look at antibacterial property, and we used Staph aureus and E. coli, and in both cases we observed excellent antibacterial activity. The zinc is the ion that causes that. So overall, we've created many composites. I'll just briefly show you a few more. This is an inkjet, which is a very unusual filler to use. Inkjet is mainly used for sintered lasering structures and for environmental purposes, not for medical applications. And we never, ever imagined that this would be the most biocompatible. So that taught us that in science, be ready always for surprises. So here we are. We've printed these composites with inkjet. You can see here how nicely porous the composite with inkjets are versus the neat polymer. And the struts were very much more well-defined when we added inkjet, and we got a lower diameter, a lower cross-sectional area, and a higher pore area. All very positive. So the inkjet proved to be an excellent filler, and here we are, primary osteoblasts growing wonderfully on these composites. So you can see not only are they growing on the struts, on the polymer struts, but also trying to jump across the polymer struts. So it's really a wonderful environment for these cells. We also looked at mineralization, because bone needs mineralization. Without that, we can't really stand up and talk like I am at the moment. So here, the red color is due to xylanol orange, and with the inkjet, as you can imagine, as you can see here, the inkjet resulted in much higher mineralization. We also looked at the alkaline phosphatase assay, and again, the inkjet proved to be the winner. So we're doing really well with these composites. Last but not the least, graphene oxide. We've created composites with that. Again, we printed these, 3D printed, FDM printed, and you can see the structures are really very, very well defined, so we can print them quite well, and the cells love them. So graphene oxide is known as it could be toxic, but if you get the right concentration, it can be osteogenic, and that's what we've proven in this work. And I'll move on to peripheral nerve tissue engineering. What happens in a nerve which is broken? It doesn't regenerate very easily. If it's a very long gap, beyond 5 millimeter, it doesn't regenerate, and the patient becomes paralyzed. So the idea here is to create a conduit like that, really a bridge between the two broken nerves, and that then allows regeneration to occur much better. So I will take you through an example. This is, I wanted to show this because this was from a collaboration in Italy, a University of Torino, Professor Stefano Goena, and Fiona is my colleague in Sheffield. So this is what we get. The nerve conduits were sutured onto broken nerves, and within about 6 weeks, look at the regeneration that we observed. And also, Stefano always told me, he is a surgeon, that it was so easy to suture your conduits. So hopefully I can take this forward in the clinic. Again, wonderful regeneration in 12 weeks. When we compared all the various parameters, like the nerve cross-section, cross-sectional area, the fiber diameter, etc. Again, wonderful, as compared to an autograft. Autograft is a real nerve you take from the animal and put it into the gap, and that worked as well as the conduit, which is really excellent for just a tubular structure made out of a polymer. And then finally, functionality. If you don't test functionality, you can't say it's worked. So we ticked the animal. It could grasp, it could dance, which is excellent. And within about 9 weeks, the autograft versus our tubular conduit were performing equally well. How good is that? So this is something I'm really hopeful, hopefully I can take to the clinic. This was another European project where we created a blend between MCl PHA, P3HO, Co3HD, which is an octanoate decanoate copolymer, with polycaprolactone, which I'm sure all of you know about. And again here, we got really excellent regeneration. I won't have time to read all the different details. If you're interested, the publication is here. But what was the good news? The good news was Neurolac is what is currently used by the surgeons, and our polymer conduit was behaving, the green one, behaving better than Neurolac. The Neurolac people didn't quite like that, but it's true. It's behaving better than Neurolac, and this is hopefully something I can take forward in the clinic. I also wanted to show you, this is a mouse that we used which has fluorescent nerves. And you can see the top showing the conduit regeneration and the bottom showing the autograft regeneration. This is a bit thinner, I agree, but as good almost as the autograft. Again, showing that our conduits work really well. I won't go into the details of this, I want to keep to time, but again, the various indices that we measure versus the autograft, our polymer conduit was even better in some cases. And this is the last, which we look at spinal cord and see whether our addition of the nerve conduit creates any pain for the mice or the rat. And we found, again, the level of pain, which was measured by a labeling marker called GFAP, was equal between an autograft and a conduit. The last thing I'd like to show you is the degradation of the conduit within 5 weeks in an animal. I've many times been challenged that medium chain length PHA actually doesn't degrade. Why do you use this? It does degrade. We have enzymes within the body like lipases and esterases that cleave them. I also wanted to show you briefly that we are now adding VEGF to our conduits, and we find it results in wonderful dorsal root ganglion regeneration. And this is again the CAM study that I mentioned, and the vascularization, and the addition of VEGF is helping the vascularization as we expect. I move on now to cardiac tissue engineering. I always kind of joke about this. This is very close to my heart, but truly it is. It's a wonderful area, and it's really challenging science-wise, and I love doing cardiac tissue engineering. So as part of a BHF Foundation center at Imperial, and what we were trying to create was a cardiac patch. After a heart attack, what happens is the heart becomes scarred. I'm sure you've fallen when you were younger and you get a scar tissue. I'm sure you remember, it's absolutely non-contractile. And imagine your heart having a scar like that. It wouldn't be able to pump the blood that you would need to survive. And what we were trying to do is replace that scar tissue with healthy cardiac tissue. How do we do that? We use iPSCCMs, the induced pluripotent stem cell-derived cardiomyocytes. I hope you remember these stem cells, which won the Nobel Prize quite a few years ago now. And we are trying to convert it to that. That is an adult cardiomyocyte. So what we did was oops, sorry, I've closed the presentation, have I? Do I just click it back? Oh, great. What we did was we carried out melt electrowriting of the polymer. And this is pretty much what we got. beautiful, well-defined structures. This is slightly better than FDM because you get much higher resolution. You can get much thinner struts with electrowriting. And what we got were beautifully mature cardiomyocytes. So Thomas Eschenhagen, who is actually carrying out clinical trial with the cardiac patches, was amazed by the actual differentiation, maturation that we managed to get. So here you can see a kind of a flipping of the color there. This is calcium transients. This is very important for heart functioning, and we got really good calcium transients on our structures. We, of course, wanted endothelial cells there as well, which allow vascularization, so we grew them on our polymer, and yes, they attached and grew, as they always do. We co-cultured them as well, and then we started putting the patch onto a rat heart. When we put it on and looked at it from a histology point of view, beautiful integration. Our bodies are very finicky. They don't like anything foreign. As soon as you introduce something foreign, they start creating a capsule around it if they don't like it. There's no capsule formation here. It's as if it's a part of the heart. So the patch was very well accepted, and then we took patches with cells and put them onto the animals, and this study is ongoing at the moment. We also did multi-material. You know, we always don't put all our eggs in one basket. If you have one material, maybe it might not work as well. So what we did was we created a patch where we added the MCl PHAs and also added alginate. And the alginate acted as the cell carrier, and the polymer acted as the mechanical stability of the patch, yeah? So this is how we bioprinted certain cells with alginate. I won't go through the whole video, otherwise time will run out. And this is what we got. Green means cells love it. They've gone through that needle, they've been printed, they still love it. So that's very good news for us that we can bioprint the cells. And we can really print the cardiac patch. So this was our printing of the healthy cardiac patch. This is just a nice image of it. And finally, and not the least, we put 3 different cell types. And this is in collaboration with a group in Spain, in the University of Navarra. And you can see here we put iPSC-CMs, which is the cardiomyocytes, cardiac fibroblasts, and endothelial cells. And these are now, as you can see, being put onto a rat heart, and we hopefully will get some positive results very soon. I move on to another new topic that we're doing in our laboratory, which is kidney tissue engineering. I know some of my family members undergo dialysis, and it's not really a pleasant thing to live where you have to get to dialysis to the hospital every single week or thrice a week. So what we are trying to do is create a filtration barrier which we could transplant into the patient, and then they wouldn't need any dialysis anymore. So we start slowly, baby steps. Firstly, does the polymer or do the kidney cells like these polymers? And yes, they do. We can see here, the red here is not dead, by the way. These cells have a fluorescent red dye, and therefore they look red. So endothelial cells, podocytes, and co-culture grew well. These are, again, printed structures. We are trying to create printed filtration barriers, and we got different angles, and all the angles resulted in high collagen production, which is the main component of the filtration barrier. We also are producing bioartificial pancreas. Diabetes is a huge problem in the world, particularly where I come from in India. Every household has a diabetic patient. So there we are. We are trying to create, again, a multi-material bioartificial pancreas, again using the medium chain-length PHA and alginate. And we find that our bioartificial pancreas interacts with glucose just as it should. So the insulin production is responsive to the glucose concentration. And here are the cells forming these balls of cells, which is how a pancreatic islet looks like. And we've also tried to add oxygen-producing calcium peroxide within the struts of the construct, because pancreatic cells need a lot of oxygen. They die because of the lack of oxygen in the human body. I won't go through everything here because I think, if I'm right, I have 5 more minutes to go. Yeah, I've got, okay. So we have antimicrobial polymers. Again, like I said, sepsis is a huge problem, more than we realize. So many deaths in the hospital because of sepsis. So we had a European project, which I led, where we tried to create antimicrobial polymers. So these are examples. We added antimicrobial peptides in this example, and we got beautiful electrospun scaffolds. We also did 3D printing with selenium and strontium doped hydroxyapatite. Again, the very nice printing activity or fidelity that we got. And I think that's all I wanted to say about antimicrobial. There's lots there, but unfortunately we don't have the time. I've also done controlled drug delivery with PHAs. We know, all of us, that drug delivery controlled is very crucial. If we don't control it, we get overdose, we get side reactions. We could even target the drugs so that we don't get any background, side effects. So this was microspheres made out of PHAs, or poly-3-hydroxybutyrate, that we used. We also made biodegradable stents. Again, a large European project that I led, and these are the stents we made. So melt extruded PHAs, cut with laser, and these are the stents. And you would ask, why would the stent, stents need to be there for some time, but why would you want it to be biodegradable? Actually, stents, they create foreign body reactions beyond a couple of years. So we really don't want them around beyond 2 years. And Abbott tried to produce one, did not succeed. So we are trying further. This is a 3D printed stent that we have. We are, of course, using FE analysis to ensure we get the right sort of structure, etc. And hopefully we'll get there soon. And last but not the least, I'd like to talk to you about bacterial cellulose, which I promised I would talk a little bit about. This is how it looks. This is an extruded polymer from, again, a bacterial culture. This is how we produce it. We produce using a bacterial culture. It simply floats up on the conical flask, and this is the pellicle, and that's the purified form. This is how beautifully nanofibrillated it is. I've not electrospun this. This is nature at its best. So it's beautifully nanofibrillated, and we've started scaling up the production using a novel bioreactor developed in my group. And actually, we have created antibacterial bacterial cellulose. So we have chemically modified them. Again, if you're interested, you can look up this reference. We have basically added alkyl groups on the surface, which then cleave the bacterial membrane. And you can see on the left, there's Staph aureus attaching and growing really well on the bacterial cellulose. And on the right, you have the functionalized bacterial cellulose, and the bacteria are attaching and lysing. They can't survive on this polymer. We are now beginning to 3D print this along with alginate. So these are now being worked on for wound healing in my laboratory. So I'd like to conclude today's talk by saying that I totally believe that polyhydroxyalkanoates are indeed an emerging class of biomedical materials. We have now scaled up the production to 100 liters. The PHAs, as you see, I've given you some examples, can be used for hard, soft and tissue engineering, for making devices, for making in vitro models, 3D in vitro models that I didn't show you today. And finally, bacterial cellulose is the new baby around the corner, and it does have a lot of promise as well. I'd like to thank all the key scientists and my collaborators without whom this work would not have been possible. I'd like to thank the funding bodies, of course, money is very important, and my group. And of course, I'd like to announce that we now have our own spin-out company. We're producing medical grade PHAs. Our company is called FAST. And we are hiring. So if any one of the youngsters in the room want a job, we have a PDRA job for 2 years and a business development manager if you've got any managerial expertise in FAST. And thanks for your attention.
Thank you very much, Ipsita, for your extremely interesting presentation. So I would like to be one of those PhD students to come to your laboratory. Actually. Now, the session is open for discussion. Any questions? Yes, there. I microfoni, per favore.
Do you want my mic?
I'm sorry, but...
No, it's okay.
No, they're coming with a microphone, so they were not ready yet, but they are coming since. I microfoni, per favore, scusate.
Opa.
Okay.
That's a very unusual looking mic.
Okay.
Yes, just. Yeah. Okay, it's funny. Don't take a picture, please. Okay, so thank you for your very impressive work. Yeah, it's very fantastic work in terms of application, biomedical applications. So your main innovation relies on the use of gram-positive bacteria, you said.
I guess the whole, all the constructs, everything is quite innovative. Not many people are making structures with them.
But in your introduction, you started by saying that gram-positive is very important because there is no lipopolysaccharide.
That was my first thing, yes.
But I have seen in your presentation the medium chain is made by negative.
Yes, gram-negative. It cannot be made by a gram-positive at the moment. And I did try. Unfortunately, I have 24 hours in a day. But one of my students, we did clone the gene from the gram-positive into the gram-negative. So we tried gram-negative to gram-positive into Bacillus subtilis. But what happened is that the pool of the substrate or the medium chain length CoAs were missing. So what one has to do is a whole metabolic engineering rather than just introduce one gene, and I pretty much didn't have the time. So what we do is we purify it well enough that there is no LPS in the final medium chain length PHA.
Okay. Any other question?
Okay, I didn't want to ask question just to play with this, just because I was really astonished by your presentation of the huge work and the excellent results you have presented.
Thank you.
Really great. I have some experience with polyhydroxyalkanoates. I was wondering whether you, thinking about high molar mass polymer, which is brittle at room temperature. When I started working with the PHB, I noticed that soon after melt processing, the material is rubbery. Then it becomes brittle with time. This means this occurs because polymer crystallizes during time, and also the amorphous parts become rigid in part. So I was wondering whether you played with crystallization by tailoring it, with trying to make it slower or faster, so to achieve the final crystallization soon after processing, or even also if you try to blend with the low molar mass polymer, which is rubbery.
Absolutely, that's what we did. So there are a few things with P3HB. One is that if you're not producing it yourself and you're buying it, it's very impure, what they sell. I don't know how Sigma sells such an impure polymer. It's blasphemy in my lab to buy PHAs, but when I moved to Sheffield, I didn't have any polymer yet, so I did use some. So I think one is that your polymer is so impure, it has other impurities that makes it more brittle than it should be. And then, of course, we have to add, like you've seen, we've been using blends all the time. So P3HB plus medium chain length PHAs form a wonderful blend. You can add very small quantities, like we did for the stent. You can add large quantities if you need. For example, electrospinning is quite impossible with just the MCl. So the P3HB makes it spinnable, and then we add small quantities. So blending is a very important property. When you're processing it, it's very important that you let it age before you do anything with it. Because if you've used it for a certain purpose, it'll get brittle over time. And therefore you will lose whatever functionality you need. So yes, A, use pure polymer. B, when you're printing, if you do printing of them, you need to be very careful about the temperature range that you use. We, in fact, that was the main thing that we did. We were very, very careful about exactly the temperature that we printed them in. We had a few other tricks in our printer that we did in order to make it work. And lastly, and not the least, is the blending with different polymers. So P3HB on its own may not do the job. Blend it with other polymers like PLLA, like medium chain length PHAs, and then you get the right properties that you need. But P3HB has some very nice properties as well, like neurogenicity, like processability. It really helps us making drug delivery microspheres. It's so easy with P3HB. Okay. Any other? Okay.
Thank you very much. Very, very exciting presentation. I have two quick questions. One is related to the biomaterial applications. Do you need to have some specific extraction techniques to remove all the impurities and to get a very good polymers for that application? And the second one is related to the possibility. What do you think of the possibility of using PHA in general to replace, let's say, common plastics, because this would mean that you need to have industrial plants that can give you a polymer that can be competitive in applications where you don't have a high added value, let's say.
Absolutely. So your first question was in terms of, sorry, I'm listening to your second question and therefore...
Yeah, the first question is related to the extraction technique.
Yes,
yeah.
Very important. Downstream processing makes or breaks the polymer. That's why I was saying this polymer that we get in the market from Sigma is so impure. Unless you purify them well enough, make sure every single molecule of, LPS or anything that co-precipitates with the polymer is very, very important. So creating a good downstream process, which is also green, is a challenge in the PHA world. At the moment, mostly we do other methods, but at the end, to get a medical grade polymer, which is absolutely pure, we do use solvents, which are not very green. And that's my thing to do now, is to create a method which is based on green solvents, like ionic solvents, eutectic solvents, to really get something that does not use chloroform, which is so difficult to get a medical grade polymer. And in terms of creating tons of these polymers, there are industries out there already, many industries, not medical grade, that's why we started fast, and they are producing tons of these polymers now. One of the biggest is called CJ Bio. They started in America, but they've moved now to Asia. So there are about almost 12 or 15 companies. There's Bluefar in China that is doing really well, but none at the moment, or not many at the moment, producing medical grade. And that's why I started fast, that we need medical grade PHAs to be brought out.
I have a question.
Yes.
Being a traditional polymer chemist, so my question is the reproducibility of the method, because it's difficult for me to understand, maybe because I have not the knowledge to how is it possible to control the process and to make it reproducible.
Very important. And that's the main reason I stick to fermentation. Even there, we have a bacteria. It has its own mind. But we ensure we have exactly the same conditions. The bacteria is stored very, very carefully because they can undergo mutagenesis if you don't store them well. So to ensure that the bacterial strain is exactly the same, so I always make sure by sequencing that I have the same bacteria every single time, every year. I sequence my strains, that they haven't changed drastically. Secondly, the pH, the media composition, the temperature, the amount of air we put into the fermentation conditions. So basically keeping all the conditions absolutely tight is crucial. And what we do get is a slight variation. So for example, let's say the monomer content going from 80% of something and 20% to 79 to 21. That's the maximum I tolerate. Anything beyond that, something's gone wrong, and I go back to the drawing table. But we haven't seen that. That's the main reason I use fermentation. So if you are a good biochemical engineer, you take care of all the conditions and the bacterial strain, substrate source, you should get repeatable polymer production.
Sorry, another quick question. The scale, what is the maximum scale up that you have
I
succeeded
have,
in?
maximum is 100 liter. That's what I have done. People have gone to tons in other industries, so it's possible. FAST will hopefully take it to that level. But fortunately, with medical grade, we don't need huge amounts like packaging and other coating sort of technologies. So I'm not aiming for millions of tons, but definitely going to 10,000 liter in the next, say, couple of years is my ambition.
Any other questions? If not, then we thank again Professor Ipsita Roy for her very interesting presentation.